Today I want to talk about the characteristic and the function of skeletal muscles and how these skeletal muscles can go to contraction. First, you need to know the functions of the skeletal muscle. One of the important functions of the skeletal muscle is body movement. The body movement means moving the bones, making facial expression, speaking, breathing, and swallowing. You know that for facial expression, you need to do contraction and relaxation of the muscles which you have in your face. For speaking, you need to do contraction and relaxation of the muscles which you have in your larynx or voice box. For breathing, you need to do contraction and relaxation of internal intercostal muscle, external intercostal muscle, and your diaphragm. And for swallowing, you need to control muscle which you have in your pharynx. The skeletal muscle can maintain your posture. They can stabilize your joint. By contraction of some muscles, you can be in a standing position or you can be in sitting position. So it can maintain your body position and your body posture. It can do protection and supportion. You know that, for example, the muscles which you have around your abdomen can protect all of your visceral organs. The muscles which you have in the floor of your urogenital system can protect and support the organs you have in your pelvis. So they can package internal organs and hold them in their own place. They can do regulation of elimination of the materials. It means that at the end of our urinary system and at the end of our our digestive system, we have some orifices. You know that at the end of digestive system, we have anus, and at the end of urinary system, we have the external opening of our urethra. And all of these openings should be protected by the skeletal muscle. So you can do voluntary elimination. Your muscle can play an important role for heat production. You know that for contraction and relaxation of muscle, we need to utilize ATP. I will talk about it next week for you, how this ATP is used in your muscles. But for this when our ATP is broken in the muscles, we have a release of heat. And the muscles can make heat and they can maintain our body temperature. You know that when you are shivering, your skeletal muscle starts to shake. By this shaking, by this movement, the ATP is broken and you can release more heat in your body. So these are the functions of our skeletal muscle. Our skeletal muscles have different characteristics. One of these characteristics is excitability. It means that they can respond to different stimulation. For this excitability, which I will talk about it more today, you know that at the end of our neurons, the somatic motor neuron, we have release of acetylcholine. This acetylcholine is released on the surface of the muscle. When it's released on the surface of the muscle, it can attach to its own receptor, and by attaching to this receptor, the channels become open, sodium ion come to our cell, and potassium ion leave the cell, so we have creation of action potential. And when action potential is created, then our skeletal muscle become excited and it can go to contraction. So one of the most important characteristics of our skeletal muscle is excitability, the ability to respond to different stimulation by changing electrical membrane potential. The other characteristic of our skeletal muscle is conductivity. It means that our skeletal muscle can receive the stimulation, but it can propagate the stimulation in the cell membrane of the skeletal muscle. It can conduct it to different parts of our cell. After conductivity, we have the other characteristic which we call it contractility. The meaning of contractility is this: our skeletal muscle can become contracted. When I start talking about the skeletal muscle organelles, you will understand it better. In a skeletal muscle, we have some microfilaments. These microfilaments are overlapping each other. When we want to do contraction, these overlapping increase and increase, and the length of our muscle decrease, and it can help our muscle to make movement. Some of our muscles at time of movement should become a stretch, and this is the other characteristic which we have for our skeletal muscle, which we call it extensibility. It means that this overlapping can become open, open, and open. So the microfilament can become far from each other. The ability of stretching of the muscle is called extensibility. And the most important characteristic after contractility and excitability is elasticity of our muscle fibers. It means that our muscle fibers, like elastic fibers, should be able to return to their original length. So this is the other characteristic which we have for our skeletal muscles. Excitability, conductivity, contractility, extensibility, and elasticity are the characteristics which you can find in your skeletal muscle. Each skeletal muscle is the organ. You know that the organ is a group of tissues which can work together to perform a specific task. In this skeletal muscle, in this organ, we have skeletal muscle fibers. The fibers are important here because they can do contraction and relaxation. We have some connective tissues which wrap our muscle fibers to protect them and separate these muscle fibers from each other. We have many blood vessels between them. These blood vessels want to bring oxygen and nutrient for our muscle fibers, and we have many nerve fibers because the nerves should bring information and bring the comment of our central nervous system to the muscles. Now I want to talk about the organization of these muscle fibers in the next picture. If you look at this picture and if you remember from a nervous system chapter, you know that each axon is covered by endoneurium. A group of axons are covered by perineurium, and they can make nerve bundle or nerve fascicle. And a group of nerve fascicles are covered by epineurium. We have the same structure, the same connective tissue for our skeletal muscle fibers too. Each muscle cell is called muscle fiber. So each muscle cell is called muscle fiber because it's long and thin. Each muscle fiber is covered by endomysium. Instead of endoneurium, you need to use the word of endomysium. A group of muscle fibers are covered by denser connective tissue, which we call it perimysium, and this perimysium can make muscle fascicle. A group of muscle fascicles are covered by epimysium from outside, and the epimysium become thicker and denser than perimysium. So from inside to outside, first we have endomysium, then we have perimysium, and the last layer is called epimysium. When we come from inside to outside, our connective tissue become more dense and more dense. And all of these endomysium, perimysium, and epimysium continue to the end of the muscle, as you see here. All of them together can make tendon. And the tendon is a dense regular connective tissue which comes from endomysium, perimysium, and epimysium, and it makes connection of our muscle to the bone. So this is the structural organization which we have for our muscle fibers. But why we need this connective tissue between these muscle fibers? We need this connective tissue because we want to bring blood vessels here, we want to carry nerves, and we want to carry lymphatics. So we need all of these connective tissue wrapping around muscle fiber, muscle fascicle, and a large group of fascicles. And all of the information are here. The three connective tissue layer wrapping our epimysium is the outermost layer, dense irregular connective tissue wrapping the whole muscle fiber. We have perimysium, which is the dense irregular connective tissue which wraps the fascicles, and it can house many blood vessels and nerves. And the endomysium is the areolar connective tissue which wrap each individual fibers. It's a very delicate layer of connective tissue, and this connective tissue can make electrical insulation. It's very important to know. It can make electrical insulation for our muscle fibers. It can do capillary support for our muscle fibers, and the other one is binding the cells to the neighboring and to the adjacent cell. So these are the connective tissue which we have around the muscles. And as I told you, the tendon is the cord-like structure of dense regular connective tissue. Pay attention, our tendon is dense regular connective tissue, but the endomysium, epimysium, and perimysium are dense irregular connective tissue. The tendon is cord-like structure, and we have the other structure which is very similar to tendon, but the name is different. We call it aponeurosis, and I talked about aponeurosis in the first chapter for you. The aponeurosis is the thin, flat sheet of dense, irregular connective tissue. And you can see the aponeurosis here. Here you can see the aponeurosis. We call it abdominal aponeurosis. The aponeurosis can connect this external abdominal oblique to the line which we have in the center or in the midline of our body. And here you can see the other aponeurosis, which can attach the internal abdominal oblique muscle to this central line, which we call it linea alba. So they are sheeted tendons, and they can connect muscle to muscle or muscle to the border of each parts of our body. So this is the other connective tissue which works with our muscle. The other structure which you need to know about it is the deep fascia and superficial fascia. And I will show you these structures in the next picture. Here you can see the cross section of the thigh. In this cross section, you can see different muscles, and you know that each of these muscles are covered by their epimysium from outside. Around the epimysium, we have the other connective tissue, which we call it deep fascia. The deep fascia is the dense, irregular connective tissue which can help attachment of one muscle to the other muscle. You know that, for example, in your upper limb, I don't know if you read this part, if you study this part in your lab manual or not. For example, in your arm, you have triceps brachii, you have biceps brachii, you have brachialis and brachioradialis. These muscles here should connect together, and they should be tightly near together. The deep fascia is the connective tissue which are located between the adjacent muscle and help this connection. After that, we have the other connective tissue, which we call it superficial fascia. The superficial fascia is the adipose tissue, and it's the areolar tissue which can connect the muscle, the group of muscles, to the skin. actually the superficial fascia is our hypodermis. Do you remember your skin is divided to epidermis, dermis, and hypodermis? The hypodermis is the adipose tissue, and you know that the adipose tissue is very connective tissue, which is located in the deepest part of the skin, and it can attach the skin to the muscles. These are the connective tissues which we have around our muscle fibers. Our skeletal muscles are highly vascularized and highly innervated because these blood vessels want to bring oxygen and nutrient, and they want to remove waste product. The skeletal muscles are innervated by somatic neuron. Pay attention, we have somatic neuron which can innervate our somatic motor neuron actually, which can innervate our skeletal muscle. And between the axon and the skeletal muscle, we have a junction which we call it neuromuscular junction. And I will show you this neuromuscular junction in the picture. And it can allow for voluntary control of contraction. Now I want to talk about the microscopic anatomy of our skeletal muscle. First, you need to know that we have some specific terms when we talk about the muscles. Inside the muscle cell or muscle fiber, we have cytoplasm, but the cytoplasm in our muscle has the other name. We call it sarcoplasm. So sarco means muscle. Sarcoplasm is the cytoplasm of the skeletal muscle. And inside the sarcoplasm, you know that we have different organelles. You have a nucleus, you have mitochondria, you have Golgi apparatus, all structures or organelles which you have in the other cells, you can find it in the skeletal muscles. Just the name of cytoplasm is changed. The skeletal muscles are multi-nucleated cells. The meaning of multi-nucleated cell is this: they contain more than one nucleus. During the fetal period, during embryonic development, we have some stem cells which we call them myoblasts. You remember that the meaning of blast is producing cell. These myoblasts combine together, fuse together, and make elongated cell. This elongated cell is one muscle fiber because it becomes long and thin. And all of these nuclei are located now in one cell. So our skeletal muscles are multi-nucleated cells. Some of these myoblasts doesn't change to muscle fibers. Some of them change to a specific cell which we call it satellite cell. And these satellite cells are located between muscle fibers. When we have any trauma, any injury, any problem for our muscle fibers, this satellite cell can do regeneration sometimes, and sometimes they can make fibrosis. Sometimes the degree of injury is high and we cannot have regeneration because we miss some of our satellite cells. And at that time, the connective tissue come and fill the gap for us. But the satellite cell themselves can do mitotic division, they can increase their numbers, then the myoblasts which are produced from them can join together and they can make muscle fibers and they can do repair and regeneration when we have any problem for our muscles. And in this picture, you can see the microscopic structure of the muscle fibers. As you see here, the muscle fibers are cylindrical in their shape. They have many nuclei. As you see in this picture, for this one, you have 1 and 2 nuclei. Our skeletal muscle cell can show these lines. These lines are called striation. So our muscle fibers are striated muscles. And this is the histological characteristic. So they are elongated and cylindrical. They have more than one nuclei. They are multi-nucleated cells. They can show a striation, and that's it. And their function is completely voluntary function. The other structure which I want to talk about it in the skeletal muscle is the cell membrane or the plasma membrane. The plasma membrane of our skeletal muscle has the other name. Instead of plasma or plasma membrane, we need to use the term of sarcolima. Sarco means muscle, and lima is the membrane of the cell. Here in this picture, you can see the sarcolima. The sarcolima or the plasma membrane of the muscle cell is very similar to the others. It's bilayer, it's composed of phospholipid bilayer, and these are the proteins which are distributed in this phospholipid bilayer. And we have the other structure here. This structure is some finger-like or some tubule structure which come inside the cell. Let me draw it here for you. Imagine that this is the cell membrane. The cell membrane makes some invaginations inside. These invaginations inside is called T-tubule or transverse tubule. And this is the T-tubule or transverse tubule which you can see here. In the sarcolima and in the T-tubule, we have many voltage-gated channels. And these voltage-gated channels, as you see here, can do conduction of electrical signals. Some of them are voltage-gated sodium channels. Some of them are voltage-gated potassium channels. And inside the T-tubule, we have the specific channel, which we call it voltage-sensitive calcium channels, which I will talk about this structure later for you. So this is the cell membrane, which we call it sarcolima. Our sarcolima makes some deep extension, deep invagination to the cell inside the cell, and it can make T-tubule or transverse tubule. In the cell membrane and in the T-tubule, we have many voltage-gated channels of sodium and potassium. And specifically in the T-tubule, we have some voltage-sensitive calcium channels. The other structure which you have and which you can see inside the muscle cell is called myofibrils. In each muscle cell or in each muscle fiber, we have hundreds to thousands of these myofibrils. These myofibrils are composed of bundles of myofilaments. And these myofilaments are wrapped by sarcoplasmic reticulum. But what is sarcoplasmic reticulum? Sarcoplasmic reticulum is actually a smooth endoplasmic reticulum in your muscle cell. So a smooth endoplasmic reticulum of our muscle cell is called sarcoplasmic reticulum. They are tunnels, they are the cisterna which wrap our myofilament, and their function is a storage of calcium ion. You know that for contraction of muscles, we need calcium ion, and these sarcoplasmic reticulum can store this calcium ion for time of contraction, and I will show them to you. Here you can see the myofibrils, and let me bring my pointer. This is the myofibril, and each myofibril is composed of many myofilaments. And the myofilaments are wrapped by sarcoplasmic reticulum. I have better picture to show them to you. We have a specific structure in our muscle fibers, which we call it triad. Now I want to talk about this triad for you. I don't know why this slide looks like this. I told you that this is our cell membrane, and we have T-tubule in our cell membrane. This red imagination is our T-tubule. On both sides of T-tubule, we have a group of sarcoplasmic reticulum. This is sarcoplasmic reticulum in one side, and this is sarcoplasmic reticulum in the other side. Now, how many structures do you have here? How many structures do you have here? You have one sarcoplasmic reticulum here, T-tubule in the center, and the other sarcoplasmic reticulum in the other side. You have three structures. These three structures are called triad. So, triad is the three structures which we have in our skeletal muscle. We have this T-tubule in the center, and it's located between two sarcoplasmic reticulum. This sarcoplasmic reticulum, as you see in this picture, can reserve calcium ion for us. The calcium ion which we have in the sarcoplasmic reticulum are binded to two proteins. One of these proteins is called calmodulin, and the other protein is called calsequestrin. When we don't have any stimulation, when we don't need to do any contraction, these calciums are bound to these calmodulin and calsequestrin. But when we need to do contraction, the calmodulin and calsequestrin release the calcium ion, and the calcium ion can perform contraction, which I will talk about it later today. The myofilament in our skeletal muscle are composed of two myofibrils. One of these myofibrils is called thick filament, and one of them is called thin filament. And now in the next picture, I want to show you what is thick and what is thin filament. If you look at this picture, this yellow structure is the thick filament. In this yellow structure, we have a protein which we call it myosin. So, our thick filament is made up of myosin molecule, as you see here. In this myosin, each of these myosin molecule is composed of two parts. These two parts have one head and one strand. Their strands or tail are twisted around each other, and their heads are in one side. And you need to know that between the head and the tail, we have the angle here. This angle is like hinge joint. Do you remember the hinge joint, like our elbow joint, can do flexion and extension? This hinge joint here is similar to our elbow joint. It can do flexion and extension here. And our thin filament is composed of three different parts, as you see here in three different colors. First, we have the red part. These red parts are glubular parts, and each of these reds is called 1G actin or glubular actin. These G actins are located one after each other, and they can make a cord. This cord is called F-actin or fibrillar actin. I will talk about G-actin later for you more. So first we have the subunits, which we call them G-actins. These G-actins are located one after each other and make a long F-actin. We have two F-actin here, which are twisted around each other, and you can see this twist here. The other structure is this green structure. The green structure is called Tropomyosin, and Tropomyosin is wrapped around the F-actins. And the other structure is blue here, which we call it troponin. And the troponin has a site here which can attach to calcium ion, which we call it calcium binding site. Now I will talk about each of these thick and thin filament more in the next pictures for you. Do you remember I told you that the sarcoplasmic reticulum can wrap around our myofibrils? This is one myofibril, and you can see these blue structures which are sarcoplasmic reticulum, and it wrap around our myofibrils, and you can see the triad here. This yellow structure is the T-tubule, and the blue on both sides are sarcoplasmic reticulum. So we have three structures, and we call it triad here. Between these myofibrils, you can see the orange structure. They are mitochondria, and they want to provide ATP for the contraction. This is the location of sarcoplasmic reticulum, which wrap our myofibrils. I want to talk about the structure of myofibrils. Our myofibrils, I want to talk about myofilaments. First, you need to know that myofibrils are made up of many myofilaments, and each myofilament is a repeating unit of sarcomere. But what is sarcomere? Look at the next picture. This is your myofibril. Each myofibril is composed of myofilament. And we have a structure in our myofilament which we call it sarcomere. Look at this picture. Can you see the green structure, then pink, then green, then pink, then green? This repeating functional unit is called sarcomere. And now I want to talk about sarcomere for you. How actin and myosin can make sarcomere, and how this structure can make contraction for our muscle fiber. The first thing which you need to know is this. Each sarcomere is composed of myosin. In the center, the myosin here is red, and the actin or thin filament is blue here. So we have myosin or thick filament in the center and two thin filaments on both sides. And in some parts, our actin and myosin overlap each other. They are located on each other. Then you can see two lines here. These two lines which can separate the first sarcomere from the neighboring sarcomere is called Z-line or Z-disc. So up to now, you know that we have myosin center. This myosin is thick filament and we call it A band. On both sides of A band, you can see only actin here. This actin is thin filament and we call it I band. And each sarcomere starts from one Z-line to the next Z-line. I told you that our actin and myosin overlap each other in some parts, But some parts which you can see in the center, your myosin is naked. It doesn't have any coverage of actin. This central part is called H zone. Here you can see H zone, and in the H zone you can see that the color is light. So if I want to talk about this electron microscopy picture, I can tell you that these two lines are called Z line. These light part on both sides are thin part or I band. This part which is darker is A band or thick filament, and in the center of A band you have a lighter part which we call the H zone, and in the middle of H zone we have a line which we call it M line. I will talk about the characteristic of each of these fibers and lines in the next slide for you. So the organization of sarcomere are A band and I band. I band is the light appearing region which contain only thin filament, and we have Z disc in the I band. The I band becomes smaller when our muscle go to contraction, and in maximum contraction we don't have I band. I'll show you the picture. And the other structure, the band, is called A band. The A band is dark appearing region that contain thick filament and overlapping thin filament. In the center of A band, we have H zone and M line. And the A band can make the central region of our sarcomere. Here you can see the structure of these sarcomeres again. Each of these structure which start from one Z line to the next Z is called one sarcomere. Let me mark it for you. Here is one sarcomere. Then you have the next sarcomere. In this part you have the other sarcomere. Each sarcomere has one central part here, which is our A band, and two parts on both sides, which are I band. In the center of each of these A band, you can see H zone, and in the center of H zone, we have M line. The H zone is the central portion of our A band. Only thick filament is present here, and we don't have any thin filament to overlap it. When we have maximum contraction, our H zone is disappear. And the M line is the middle part of our H zone. It's a protein meshwork which can be a site of attachment for our thick filaments. This is the bigger picture. In this bigger picture, you can see the A band. The A band is located in the center. In the A band, we have myosin, and we have some overlapping actin here. You can see two I bands on both sides. Your I bands, as you see in this picture, contain only actin. We don't have any myosin in this part. You have some spring-like structure. They are proteins which we call them connectin. Let me highlight it for you here. We call them connectin, and these connectin, as you see here, help attachment of our thick filament to the Z disc. The Z disc are the limit of our sarcomere. The parts of your thick filament or A band which we don't have any actin in the center is called H zone, and in the center of H zone you can see this line which is M line. This is the structure of sarcomere. Here you can see myosin or thick myofilament. The thick myofilament or myosin is very similar to golf club. As you see here, it has head and it has tail. These two golf clubs are twisted around each other and they can make one myosin molecule. The myosin molecule has two heads and two tails. Between head and tail, we have hinge joint, and this joint is very important at the time of contraction. Each head of myosin have a site for attachment of ATP. ATP can attach to the head of myosin. The head of myosin have the characteristic for breaking the ATP down. So it has ATPase activity. And by this activity, the ATP is broken, we have ADP and phosphate, and the energy is released and heat is released. The energy is for the contraction of the muscle. So this is about the myosin. And in the next picture, you can see actin. I told you that the actin or thin myofilament, it has three different regions, and you can see them by three different colors. First, we have this glubular actin or G-actin. Look at here, each G-actin contains one small yellow part. These yellow parts which you have here is called active site of actin. This active site should attach to head of myosin at the time of contraction. Then these G-actins are located one after each other, and they can make fibrous actin or F-actin, two strands of F-actin twisted around each other as you see here. The next part is this blue part, which is tropomyosin. Tropomyosin is the elongated protein which wrap around our actins, and this wrapping is not accidentally. This wrapping is in such a way that all active site of our actin is covered by tropomyosin. So the active sites are not naked. And then we have the other molecule which we call it troponin. The troponin is composed of three different subunits. One of them is called TNA. The TNA is a part which attached to actin, F-actin. One of them is called TNT. This is the TNT which can attach to tropomyosin, and the part which is located in top is called TNC. The TNC is the part which is attached to calcium ion. So we have three different subunits for troponin. Again, here you can see the thin filament or actin. In actin, we have two F-actins. Each F-actin is made up of many G-actins, and each of our G-actins have one active site, the yellow dot here. These active sites are covered by the other protein, the strand-like protein, which is tropomyosin, and then last subunit is called troponin. And our troponin is composed of three units. TNA, A means actin, so it's attached to actin. TNT, T means tropomyosin, so this part attach and binds to tropomyosin, and the other one is TNC, which binds to calcium ion. And here you can see a sliding filament model. Look at here. Here you can see the relaxed sarcomere. In relaxed sarcomere, you have two Z line on both sides. You can see I band or light band on both sides, and A band, which is dark, in the middle. And in the middle of A band, again, you have a lighter part, which is H zone, and you can see M line. When your muscle go to contraction, when it's partly contracted, you see that the length of our sarcomere becomes shorter. And here in partially contraction, you can see I band, but the H zone is very, very small and short. And in fully contracted, you see that we don't have any I band and we don't have any H zone. So these fibers slide over each other at the time of contraction. They overlap each other. So at the time of contraction, our I band and our H band become disappeared. We have two proteins, which I talked about one of them for you in the sarcomere. One of these proteins is called connectin. Connectin is the spring-like protein which can attach our thick filament to the Z line. It extends from the M line to Z line, and it can help keeping the organization stable. And the other protein is called dystrophin. Dystrophin is the protein which anchors some myofilament to sarcolima protein. And the abnormality of this dystrophin can cause muscular dystrophy, and this is a disease which we have specifically in female. We call it muscular dystrophy. In this problem, in this disease, we have insufficient or defective dystrophin. The sarcomer damage during muscle contraction, and calcium enter the cell and damage the protein. This problem begin at the time of childhood. We have walking difficulties, muscle atrophy, and we have some postural issues. This disease cannot be treated because the dystrophin inside each muscle fiber is damaged, and because we cannot have contraction and relaxation specifically for our respiratory muscles, we have dead for this patient, and they cannot live beyond age 30. The other structures which we have in our skeletal muscle is our mitochondria. Here, in your book and in most of the textbook, it writes mitochondria, but you need to know that the name of mitochondria in the skeletal muscle is sarcosome. So sarcosome is the name of mitochondria. These mitochondria can make energy. The myofiber have many mitochondria because they want to make aerobic ATP production. We have myoglobin in our muscle. The myoglobin is a protein which can attach to oxygen, and this oxygen is very important for making ATP for us. We have a large storage of glycogen in our muscle cells, and it works as a fuel of the muscle. And we have the other reservoir for our muscle contraction, which we call it creatinine phosphate. I'll talk about this creatinine phosphate next time for you. It can quickly give up its phosphate group, and it can make ATP for us. I will talk about it completely for you. So you need to know that we have many mitochondria. We have myoglobin. The myoglobin is responsible for the red color of your muscles, skeletal muscles, and it can attach to oxygen. You need to know that if this protein is located in our blood cell, we call it hemoglobin. And in the hemoglobin, it has the same characteristic. It should carry oxygen. The glycogen and the creatinine phosphate are the fuels which we have inside the cell, and they help our skeletal muscle cell to provide and make more ATP for us. The next structure which I want to show you is the motor unit. The motor unit is the nerve, the motor neuron, the somatic motor neuron, and the muscle fiber which is controlled by this neuron. So the combination of the somatic motor neuron and the skeletal muscle is called motor unit. We have two types of motor unit. One type of these motor unit is called a small motor unit. One of them is called large motor unit. A small motor unit means that we have one somatic motor neuron, and this somatic motor neuron come and innervate less than 5 muscle fiber. Less than 5 muscle fiber. So it can control each muscle fiber better. It can allow us to precise control of force output. For example, if you want to do something very neat and precise with your fingers, when you want to move the muscles around your eyes, you use this motor unit, a small motor unit. But we have the other motor unit, which we call it large motor unit. One nerve come and innervate thousands of muscle fibers. We don't need that much control for these muscles. For example, the muscles which can make your trunk, like external abdominal oblique, internal abdominal oblique, transverse abdominis, rectus abdominis, all of the muscles are controlled by large motor units. One nerve come and innervate a group of muscles. So this is the meaning of motor unit for our muscles. And the next structure is called neuromuscular junction. I don't know why this slide looks like this, but I will talk about this slide in this picture. This is the close-up of neuromuscular junction. In the neuromuscular junction, or NMJ, you can see the axon terminal. Let me show you the axon terminal here first. This is your axon, and this is the synaptic knob or axon terminal. In our synaptic knob, we have many vesicles, and these vesicles contain neurotransmitter. Then you can see a synaptic cleft here, and after that you have postsynaptic membrane. The postsynaptic membrane in our muscle, skeletal muscle, has a specific name. We call it motor end plate. So the postsynaptic part is called motor end plate for our skeletal muscle. And as you see here, it's ruffled. The border is folded here, and I will tell you why. The information comes from the axon to the axon terminal. When the information comes to this part, the calcium channels become open. Calcium ion come to our cell, attach to the vesicles which contain neurotransmitter, and help these vesicles to do exocytosis. Exocytosis take place, and the neurotransmitters release in this synaptic cleft. Then the neurotransmitter should attach a receptor on the motor end plate. They attach to the receptor. When they attach to the receptor, they can make excitation or action potential here. It means that some sodium channels become open, sodium ion come to our cell, potassium ion leave our cell, and this is the meaning of action potential. So this is the neuromuscular junction. And I told you that we have folded surface for our motor end plate. When our surface become folded, the surface increase, and we can hold more receptors here. This is why do we have more surface here. We increase the surface by this folding. And you had one question in your practice questions of chapter 23 of your lab. And this question asks you about if we don't have cholinesterase enzyme here, what happened to the muscle? You know that when the neurotransmitter release in the synaptic cleft, when it attached to the receptor, the extra neurotransmitter should remove from the synaptic cleft. Sometimes we have diffusion. Sometimes we have uptaking of this neurotransmitter to our cell, to our neuron, and the neuron can recycle this neurotransmitter. And sometimes we have enzyme like cholinesterase or acetylcholinesterase, which should digest and break down these neurotransmitters. Because if neurotransmitter remain here, they make hyperstimulation of our muscle, and our muscle cannot go to relaxation anymore. So we need them here. And if you look at this picture, you can see the muscle fiber and the neurons which come here. This is our somatic motor neuron. You need to know the type of neuron here because your skeletal muscle can work voluntarily under your control. So the somatic motor neuron should innervate it. This is the synaptic knob here. It contains many synaptic vesicles, and this structure is called neuromuscular junction. You have synaptic cleft and motor end plate here, which is folded and contain many neurotransmitter. So when the neurotransmitter is really here, attached to its receptor, when the sodium channels become open, we have depolarization of our cell, and the wave of depolarization should propagate in our sarcolima. The action potential which happen here propagate in our sarcolima until it reach the T-tubule here. When it reached to the T-tubule, it come inside the T-tubule. Do you remember in the T-tubule we have some sensitive voltage gated calcium channels? Then these sensitive gated calcium channels can open the calcium channels of our sarcoplasmic reticulum, and calcium ion release in the cell. When the calcium ion release in the cell, the calcium can attach to the TNC of troponin. And then we have some configurational changes, and then the actin and myosin can overlap each other, and they can make contraction. I will talk about this property in the next session for you.